Well, welcome everyone to our September edition of the Coastal Coral Seminar Series. Just before we begin, I'd just like to make an acknowledgement of country and just acknowledge the traditional owners of the lands on which we meet tonight. So for all of you, wherever you might be, I'm personally in Minjerribah. And I'd just like to also extend that acknowledgement to any traditional owners and Indigenous people we might have with us joining over Zoom tonight. So I'm sure all of you are aware, but we're here for a talk by Dr. Nataly, and she'll be talking about reproductive biology of corals. But before we get right into today's talk, I'll just give a brief overview of Reef Check and highlight some of our upcoming events for those that might be interested. So for anyone that doesn't know, Reef Check is a global organization, and of course we're meeting as part of the Australia branch of that organization. And it's really all about protecting our reefs, our oceans, and empowering people to do so. So that's through both engagement and outreach opportunities and events, as well as through our worldwide citizen science program. That's aimed at collecting long-term monitoring data from various reefs. For us, that's mainly in Southeast Queensland and along the Great Barrier Reef. So just before we begin tonight, just some housekeeping. Please feel free to keep your video on if you'd like, but please keep yourselves on mute while Nataly's speaking, of course. If you want to ask any questions, feel free to pop them in the chat. At the end of the session, we'll extend some question time, and you're welcome to either put those in the chat. You can either message me personally or the whole group. That's all good. And if you want, you're also welcome to unmute yourself and ask the question directly. That's all good. If anyone isn't on our mailing list currently, I've got the email up there on the slides. Feel free to jot that down and join our mailing list. As you'll get information about all our upcoming events through that mailing list. And just before we get started, I'm going to attempt to do a group photo. So if everyone wants to turn their videos on, if you don't want to, that's absolutely fine as well. My screenshotting is not working, so I might have to use my phone. Alrighty, everyone smile. Awesome, thank you. In the absence of being able to meet in person, that's the best we can get. So for our upcoming events, if anyone's interested in becoming a reef surveyor, so as I mentioned previously, a huge component of Reef Check is our citizen science program where we collect data in water. So that's primarily on scuba. If you are interested, we have some training sessions that have been broken up into an online component, and then of course there's the in-water practical component. So that's going to be running on the 15th, 22nd, and 29th of September. If you'd like more information or clarifications, feel free to email Jenny. So her email is at the bottom there. This is also on our Facebook page. And of course, you're welcome to screenshot this as well, take a picture of it if you think you might forget. We also have the Calandra Music Festival raffle, which is coming up. So that's supported by CMF. And it's really awesome. You can win a family pass for 2 adults and 2 children for 3 days, and that's valued at $850. So to do that, just make a donation to Reef Check Australia of $25 or more from now until the 26th of September. So get in quick since we don't have that many days left. And on the 27th of September, the lucky winners will be drawn. We also have a great beach cleanup happening at Forrest Beach. So that'll be on Sunday, the 2nd of October. and that's open to anyone. So you don't have to be a Reef Check ambassador or surveyor to join in on that. So that'll be in the early mornings, so 7:30 to 9:30 at the Forrest Beach boat ramp. And I'm sure there'll be many people keen to stay at the beach afterwards or catch up over coffee. So it'd be a really great way to learn a bit more about Reef Check or get in touch with other people and just meet like-minded people at that event. So before I hand over to Nataly, I just want to introduce next month's speaker. So that'll be Dr. Vicky Martin from the School of Earth and Environmental Sciences at the University of Queensland. So she's a social scientist, and she'll be talking about some of the work she's been doing locally, or at least locally for myself, and that's in Quandamooka and Moreton Bay. And she'll be talking about social values and perspectives, primarily of fishers and people that use the area and the bay for recreational use. And of course, if you want to get involved or you're interested in any of the events at all that I talked about, or if you'd like to become an ambassador like myself, or a Reef Check surveyor, please don't hesitate to get in touch with any of us and get involved. We're always looking for more people, particularly surveyors and ambassadors. So the easiest way to do that is either through our mailing list, which I mentioned earlier, or feel free to add us on any of our socials. So we're across Facebook, Instagram, Twitter, and LinkedIn. So we're really easy to get a hold of. And just before we move on, I'd just like to thank all of our sponsors and everyone that made this night possible. It really wouldn't be possible without many of our gracious sponsors, particularly the Sunshine Coast Council and City of Gold Coast, as well as the Port of Brisbane. And of course, thank you to all the other volunteers, and particularly Jody and Julie, who really are the reason that many of these events are able to happen. They're really sort of our champions. So over to Nataly. So Nataly finished her PhD, I think it was last year, and she worked primarily on coral larvae and reproduction in corals. She's now a postdoctoral researcher at the University of Queensland. She has a passion for coral reefs and coral reef restoration, and is particularly interested in understanding reef dynamics, reef reproductive biology, causes of coral reef degradation, and traits involved in coral species adaptation. Her PhD was focused on some reproductive aspects of coral species and coral offspring performance under experimental conditions. Particularly, she explored larval response to changing thermal conditions, which could shed light on understanding the future of some coral species coping with climate change. So over to you, Nataly. I'll just stop sharing and you're welcome to take over.
Thank you, Ilya, for that introduction. Let me see. Hopefully everything works fine. Hello, everyone. I would like to thank you for joining me tonight for this presentation. My name is Nataly, and today I'll be talking a little bit about coral reproduction. But first, I would like to acknowledge and pay my respects to the elder past and present, to those who have passed before us, and to the members of the Aboriginal and Torres Strait Islander community who may be also listening to this online presentation. Second, I would like to give a brief introduction about coral reefs. I'm aware that maybe many of you living here in Australia and attending calls to coral talks or similar talks already know about these magnificent ecosystems, particularly have heard about the Great Barrier Reef and the bleaching events that have affected this ecosystem in recent years. However, I would like to make sure that we all are in the same page related to the importance of coral reefs and threats they're facing before I continue to talk about coral reproduction and some of my work. So coral reefs are subaquatic structures made of calcium carbonate deposits, mostly composed of the skeletons of colonial marine invertebrates called corals, but these calcium carbonate deposits also can come from other organisms such as calcareous algae and mollusks, among others. Here, we have an example of one of the most well-known and largest coral reef systems on Earth, the Great Barrier Reef. The Great Barrier Reef, or the GBR, which is easier for me to pronounce, makes up 10% of the world's coral reef ecosystems. It spans over 2,300 kilometers from the Torres Strait just south of Papua New Guinea and down the eastern coast of Australia. It covers roughly 348,000 square kilometers in area, and it is made up from about 3,000 individual coral reefs, and overall is a quite complex, unique, and breathtaking ecosystem. Moving to more generalities of these ecosystems, we know that most coral reefs thrive in warm, tropical, shallow sea waters, and they can generally take the form of a fringing reef, a barrier reef, or an atoll. So fringing reefs is the most common type of reef. These type of reefs are found around a landmass. They form borders along the shoreline, and they also surround islands. Barrier reefs are similar to fringing reefs in that they also border shoreline. However, they are separate from land, which creates a lagoon of open and often deep water between the reef and the shore. Finally, the other common type of coral reefs is atoll. These happen when a fringing reef continues to grow outward from a volcanic island that has sunk below sea level, and atoll is formed, and then an atoll is formed. Atolls are usually circular or oval in shape with an open lagoon in the center, or as we can see here, they can have the Superman logo shape, which is pretty cool. So why are coral reefs important? Coral reefs are some of the most diverse ecosystems in the world. These underwater structures provide homes to an enormous number of other marine organisms. Actually, many of us have heard that reefs are often called the rainforest of the sea. And this is due to the high diversity of life found in the habitats created by corals. More interesting is that although coral reefs only cover a small percentage of the seafloor, which is about 0.2%, they support at least 25% of all marine life. So fishes and other marine organisms shelter, find food, reproduce, and rear their offspring in the complexity of these underwater structures formed by corals. Coral reefs also provide multiple services that directly and indirectly benefit humans. Coral reefs protect coastlines from storms and erosion, provide jobs for local communities, and offer opportunities for recreation. In general, over 500 million of people depend on reefs for food, income, and protection. And coral reefs are also a source of new medicines, considering that a lot of research to develop new drugs is focusing on marine organisms. In the case of Australia particularly, the Great Barrier Reef is an economic powerhouse, contributing more than 6.4 billion dollars each year to the Australian economy and around 64,000 full-time jobs. There are a number of factors threatening coral reefs around the world. Coral reefs are affected by pollution and sedimentation, overfishing, and in general, any destructive fishing practices such as dynamite and cyanide fishing. Coral reefs, and particularly corals, are also susceptible to multiple diseases, and of course, bleaching, which is caused by ocean warming. And considering that coral reefs provide a lot of ecosystem services, if they disappear, essential food, shelter, and spawning grounds for fish and other marine organisms will cease to exist. Biodiversity will greatly suffer, economically important species will disappear, and food and income of local communities will also be affected, among other negative effects, like not being able to dive in such a beautiful places. Now that we talk about coral reefs, their importance and threats, let's talk about corals. The corals that build reefs are known as heart or reef building corals. These are colonies of hundreds to thousands of tiny individuals called polyps. And polyps are in charge of secreting layers of calcium carbonate to form their hard exoskeleton. Importantly, coral colonies are sessile, while when they are adults, meaning they're fixed to the substrate. And another feature is that different species have different ways of building their skeleton, leading to a wide variety of coral growth forms or morphologies that will increase the three-dimensional complexity of coral reefs. As we can see in the figure at the bottom, there are 5 main coral morphologies: encrusting, hemispherical or massive, tabular or plate-like, corimbos, and branching. And these different forms will provide distinct advantages and disadvantages when coral species are competing for space and light. Now, finally, coral reproduction. Let's talk a bit about it. Corals have evolved a remarkable range of reproductive strategies to survive in their dynamic environment. For example, corals can reproduce asexually and sexually. Asexual reproduction generally leads to coral colony growth. As we can see in the pictures, coral budding or division is a way of producing new polyps, either from parent polyps, as you can see from the picture on the left, or new polyps can be formed outside the polyp walls, as you can see in the picture on the right. Another form of asexual reproduction is known as fragmentation. Fragmentation occurs naturally when coral pieces are broken off a colony as a result of wave action, storms, or animal activities. Under favorable conditions, these fragments can attach and develop into new colonies, attach to the substrate and develop into new colonies. And thanks to this feature, some initiatives to help coral restoration after human disturbances, such as boat grounding, include rescuing coral fragments and taking them to coral gardens where fragments can have a chance of regrowing. As I mentioned before, corals, adult corals, are sessile. Therefore, sexual reproduction plays a key role in coral dispersal, which mostly relies on mobile propagules. Moreover, sexual reproduction is also key to maintain genetic diversity of species. In terms of sexual reproduction, there are two main modes of reproduction: brooding and spawning. Brooding coral species have internal fertilization and release developed larvae that are competent to settle, like the one shown in the picture at the bottom. Broadcast spawners can be either gonochoric, meaning some colonies are male and some colonies are female, or hermaphrodite, meaning colonies produce both male and female gametes. And these broadcast spawners usually release their eggs and sperm in mass spawning once a year, or this can vary from region to region. And well, this spawning can be observed as spawn slick on the surface of the ocean, as we can see in the picture at the bottom right. Then the gametes that were released drift to the water surface where fertilization takes place, and commonly after a few days, the embryos will have developed into the coral larvae. So now I'm going to introduce some of the results from my research about the effects of temperature on coral larval performance. This was research that I developed as part of my PhD at the University of Queensland. The the reasons behind this research are related to some of the coral threats that I already explained. We know that climate change is modifying species populations with the potential to disrupt ecosystem functioning and reduce biodiversity. Considering that global warming is a main threat, knowing that thermal tolerance of species might shed light on species potential, knowing that thermal tolerance might be important to know species potential to adapt to climate change and particularly to ocean warming. Ocean warming is a strong selective force on coral species, creating changes across tropical and subtropical reefs and jeopardizing their persistence. Moreover, subtropical environments may act as refugia for tropical species, or this is our hypothesis that once the temperatures increase to a point where species cannot survive in them, they will migrate to areas where temperatures are lower. However, the potential that subtropical reefs have as refugia for coral species relies partially on larval dispersal and the distances that corals are allowed to cross and reach these refugia, as well as on larval performance at cold conditions, because currently these cold conditions in subtropical environments are outside the thermal range limits for tropical coral species. So as I mentioned before, most coral synchronize the releasing of their gametes into the water column following different cues. These cues could be changes in water temperature, moon phases, tides, among others. Once in the water column, fertilization and larval development occur, and during this stage, coral propagules get dispersed by ocean currents. When larvae become competent or start swimming, they search for suitable substrate where they can settle and eventually form new colonies that reach sexual maturity and keep the cycle going. Therefore, I was interested in studying dispersed larvae from tropical corals that must survive the pelagic stage as well as maintain the ability to settle to colonize subtropical reefs and establish coral populations. Considering that determining the effects of temperature on the early life history stages will improve prediction on how species are responding to climate change, we aim to understand how coral larval performance could be affected by temperature, particularly survival of the larvae in the water column and settlement rates or success of these coral larvae. This study is placed in the context of ocean warming. It also requires like the southward transport of larvae from the Great Barrier Reef by the East Australian Current and the predicted increased influx of tropical species into subtropical coral habitats that may, as I mentioned before, may act as a future climate refuge. So we aim to answer if different temperatures affect larval performance measured as survival and settlement rates. And we also wanted to know how other factors such as species and pelagic larval duration that I also gonna refer to this pelagic larval duration as larval age for this study. So how species and pelagic larval durations will interact with the potential effects of temperature on larval performance. We ran 2 experiments simultaneously and evaluate the effects of temperature, species, and pelagic larval duration or larval age on larval performance. Particularly, we analyze the variation of survival and settlement rates. We use 4 species of Acropora, which is a genus that is an important reef builder and that confer a lot of structural complexity to the reefs. And we ran survival experiments for a maximum of 24 days and settlement experiments for a maximum of 26 days. So as you can see in the picture, we collected the coral colonies and kept them in aquaria until they spawn. So that was right after full moon, checking every night for signs of spawning. Once they spawn, we facilitate fertilization and raise larvae until they were competent. So as you can see in the picture, there were lots of bowls and Nataly beans containing millions of coral larvae that we raised, so we were able to run the experiments. Finally, when the larvae were competent, we set up our experiment. As you can see in the picture, there are like 3 main areas. So in red, they're the small containers where we run the survival experiments. Over here, there were some containers where we run the settlement experiments. And over here, there were just reservoir containers having larvae, so we could restart the settlement experiment over time, 4 times. So as results, the probability of larval survival depended on the interaction between all the 3 variables, meaning temperature, species, and larval age. overall species and larval age were the main variables influencing survival, while temperature had a less pronounced effect on larval survival. The figure shows on the x-axis the pelagic larval duration or larval age. So here. And in the y-axis is the probability of survival that could also be think as a percentage if we multiply these numbers by 100. So as a general pattern, survival decreased over time for all species and temperature treatments. However, there were some differences depending on interactions with species. For example, although survival at the first sampling point was about 75 for most species, however, the hot treatment of Acropora digitifera and the overall survival probabilities for Acropora hyacinthus were significantly lower. Interestingly, Acropora digitifera and hyacinthus reached a 50% probability of survival before the other two species, which could indicate that they are more susceptible to larval age. Moreover, the probability of larval survival dropped below 15% by a larval age of 16 to 17 across all species. In terms of settlement, the probability of larval settlement depend on the interaction between temperature, species, and again, larval age. Overall, species was the main factor influencing settlement, followed by larval age, while temperature had a less pronounced effect. This figure shows pelagic larval duration or larval age on the x-axis and probability of settlement on the y-axis. And the temperature treatments are differentiated by colors. The probability of larval settlement was below 75 or roughly below 75% for all species, irrespective of temperature or larval age. Settlement probability increased over time, leading to a settlement peak for larvae of 3 species, well, 2 species, Valida and Spatulata, and potentially Hyacinthus. We didn't have more sampling times for Hyacinthus, so we don't know if there is a peak or if the pattern keep going. In contrast, settlement probability of Digitifera decreased gradually from the first sampling point, from around 40% at a larval age of 9 days old to around 12% at 15 days old. So with not strong influence of temperature overall. To summarize, larval performance was mainly affected by species and larval age, such as larval survival decreased over time for all species, but the survival was strongly species specific. Settlement also varied considerably among species. We also found that the effects of temperature were species and larval age specific, depending on mostly depending on the larval age. Importantly, our experiments started when larvae show substrate searching behavior or when they were swimming actively. So the results, although there are results of some studies that show susceptibility of swimming larvae to changes in temperature. Results indicate that temperature did not have a strong effect on coral larvae, not performance. However, our study does agree with other research that have shown that swimming coral larvae tend to be less sensitive to thermal variation than potentially earlier life stages, such as the embryogenesis and larval development. Considering the speed of the East Australian current, some models indicate that acropore species have the potential to be transported away from their natal reef, in this case, Heron Reef, and reach subtropical coral communities as far south as the Saltry Islands within a range of between 8 to 18 days, which may imply that only a small proportion of larvae will be able to reach these distant communities, given that larval survival probabilities decrease below 50% for larval age over 9 days. So some key points and potential implications. Temperature doesn't have a consistent effect on larval performance of swimming larvae. Instead, its effect will depend on species, their specific thermal tolerance of species, and how this thermal tolerance varies over time or with larval age. In our study, larval longevity plays an important role for both survival and settlement, and is key for larval dispersal. Our study also shows that once development is complete, temperature had little effect on survival and settlement. It is important to consider that our results refer to swimming larvae, and this is a key distinction that we need to keep in mind when analyzing our results, because it has been shown that increased sea surface temperature negatively impacts fertilization rates and embryonic development, and that survival will decrease over time. Therefore, temperature has an impact in coral migration, which can still, even if there's still some migration, this could occur gradually due to the high mortality during the early development stages. And we cannot forget like local retention dynamics due to currents. Overall, we still think that including survival as well as settlement is key to improve prediction of models of species responses to climate change. Ideally, we would like to have a study that not only covers 20-something days, but it goes from all the way from fertilization to maybe try to increase experimental times up to 2 months or 3 months and see what happens with survival once the corals have settled. Finally, as with any research, this was a collective effort where a lot of people were involved and helped to make this work possible. And open to questions.
Thank you, Nataly. That was really awesome. We'll just give everyone a bit of time to put their questions into the chat, but I can see that there is one already there. And that's from Marie. The question is, how long do dispersal corals remain viable on the Sunshine Coast? And does this happen on full moons?
Well, I don't know specifically about the Sunshine Coast, but coral larvae have, well, progressively survival of coral larvae is decreasing But of course, there are studies with running experiments with some coral larvae that have kept coral larvae alive for up up to 3 months or even longer than that. But I will say that that's the exception more than actual the rule, because there's also a point where the coral larvae don't feed from the water column, so they're depleting the nutrients while they're in the water column. So in some point they need to settle, and they probably, even if they could stay longer in the water column, they're probably gonna opt for settle, search for a substrate and settle. And the full moon question, like corals, corals are quite diverse in the way they reproduce, but when we're talking about broadcast spawners, that are the ones that release female and male gametes into the water column, they usually do it once, maximum twice a year. It's gonna depend on the region. And the main cue that is think that leading the spawning is changes in water temperature. So in Heron Island, for example, and the Sultry Islands occurs when the water temperatures start increasing. So at the end of spring, early summer, in Sultry Islands takes a little bit longer. So it starts, there's some sort of delay because the water is colder over there. So they start in January, more or less. And then the second cue that is quite important is the moon phases. So after a full moon, we start like counting nights. So for some species, it's sort of well known that between 7 to 9, 10 nights after full moon, you're gonna find like they're gonna release the eggs if they have them. But that will be again located in these seasons after, like after spring, early summer. I don't know if that answer.
I think so. There's a few more. So, is there dispersal of corals to the north?
Sorry, can you repeat that?
Is there dispersal of corals to the north? So,
Well, it's gonna depend on ocean currents. So yes, there's definitely in the Great Barrier Reef, you can see that there like a connection among coral reefs that are close to each other, and that's gonna vary depending on the ocean currents and conditions. Because although I use the word swimming larvae, doesn't mean they're not professionals. they mainly transported by ocean currents, and their swimming is probably more gonna influence the vertical swimming in the water column when they start searching for substrate to settle. But yes, there gonna be movements in between coral reefs, and that's gonna be uh, lead by ocean currents and the patterns.
yeah. We have another one, and this is from Ruth. She says, very nice presentation, thank you. Do you think that larvae of other coral species would have the same sensitivity to temperature?
Uh, I think so. Uh, Sadly, with experiments and with coral reproduction work, we end up working a lot with Acropora genus, because is like the spawning is well known, and you almost have for sure, if you collect colonies that have eggs, you know that you're gonna get the larvae, and they're sort of easy to um, rear and to work with afterwards. But I think that the tendency of being susceptible to temperature is applies to all species, different types of organisms. So I will say that yes, other corals are going to be also susceptible to temperature, but uh, it is always important to like increase the data and be able to capture like the variability that it could exist between the with among different species.
Yeah. So we got quite a few questions coming in here. So another one from Hassan. Do aquaculture activities in coastal areas, for example, affect corals?
Well, I like that everything is gonna go down to the management of the water and the waterways and all the like waste in general from these activities. I will say yes, it is really difficult to control in water, like even if it's a if you try to control something, but if the aquaculture is happening in these areas, it's really difficult to control what is leaving this area and affecting like the coral reefs nearby. So I will say yes, but I guess that management does a big part of the job to try to keep that to minimal.
Yeah, sure. And Lily is asking, were any massive corals included in your study? In Western Australia, we've had some bleaching events recently, leaving only massal corals alive, and most branching corals are bleached or dead. Would you suggest the resilience is physiological or genetic?
I didn't run any genetics on my samples, so I cannot have a good grasp or what's the main mechanisms like affecting that. But again, sadly, I didn't include massives. I have data from massives about spawning, but for the numbers that I require to run my experiments, and also because like the idea was like reducing to the minimal other factors that could like be influencing the results of my experiments, so I couldn't include. I think I started with a list, a huge list of potential species and the factors that I wanted to compare. So see branching versus massive. And yeah, after a couple of meetings with my advisor committee, they helped me to like try to focus, and it's like this is not gonna work. I'm not sure if no, I don't know about like similar. There are some experiments with uh, other species, some of them including massives. But yeah, no, it wasn't part of um, this research.
Awesome. So we have one from Lowell. I would like to ask if other life forms of corals or species have the same response to changing temperature.
Um, there, well, corals are really like when we use the word corals, we are accounting for a different and great uh, diverse group. So we also talk about soft coral, and like there are corals that uh, contrary to the ones that we usually refer as reef builders, they don't have symbionts, the zooxanthellae, the algae that lives in the coral tissue. So we know that the coral bleaching occurs because rising temperatures disrupt the relationship between the algae in the coral tissue and the corals. So the coral algae is expelled by the corals, and that's why they lose the color, and that's why they look white. And depending on how long those stressing factors last, the corals could eventually die. So taking that as an example, if we look at corals that don't have algae as symbionts and they don't rely on that symbiotic relationship to survive, they potentially will be less susceptible to the increases in temperature, or at least it wouldn't affect it in the same way that the reef builders are affected.
Yeah, awesome. I think that explains it pretty well. We have another question from Marie. Do the ocean fish and creatures eat the coral when it spawns? I ask because around this time I see a lot of activity when it happens. Thank you.
Oh yeah, that's definitely of festivities for a lot of marine organisms around corals when they spawn. Yeah, it's a great source of lipids and energy. So yeah.
Yeah, yeah, for sure. It's always a frenzy at spawning.
Yeah.
I'm just gonna have a look and see if there's more questions. I've also noticed a few people asking about recordings, just to let you know, this talk is recorded and it'll be up on the Reef Check Australia YouTube channel in a few days' time. So it'll be publicly available if anyone missed it or wants to rewatch it. Cool. So we have another question down here. Thank you for the presentation. The information demystified many of the statements I hear in the industry about climate change impacts to the corals of different reefs within Australian marine parks. I will include the explanation of this study in my guided walks to tell my guests about the amazing strength of our reefs. That's a really cool comment. And Lowell's got another question. What life forms are able to spawn aside from branching corals?
The from the different morphologies that I shown, that again they're just main uh, 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 morphologies or shapes, but they're like a bunch of different mixes between among all of them. Uh, all of them have spawn, like all of them have the capabilities. It's gonna vary from uh, mostly between genus and families. So some families or some genus are known as brooding species. That means they release the sperm. So all the female colonies collect that sperm, and they're able to do the fertilization internally. So what they release at the end is the larvae instead of the gametes. Uh, but uh, other species are gonna be, as I mentioned, either like the spawners, and yeah, like all of those morphologies have the potential to release the gametes in the water column.
Yeah, great. I think that's all the questions for now. So thank you again for your awesome talk. And I think just the number of questions we received is just an indication of how much everyone enjoyed it. So just for everyone that's still with us, Nataly and I will still hang around for a couple more minutes. And if you have any questions at all for Nataly, or if you want to ask any recheck related questions to myself, feel free to hang about. We'll probably just stay on for another 5-10 minutes, depending on how many of you decide to stay on. But otherwise, feel free to hop off the meeting if you'd like to have dinner or head off for the night. And I'm sure Nataly would appreciate some thank yous in the chat for her awesome effort tonight. But thank you again for joining us, everyone. And please follow Reef Check on all our socials and keep in touch and eye out for next month's event. Thank you.